Thanks, Jason. Yeah, I think the title and abstract got kind of messed up. So this is the actual title, and don't worry about the abstract. It's somewhat different. Anyways, it's a pleasure to be here, and I really enjoyed many of the presentations this morning, so it'll be a tough act to follow. But I'll try and introduce the concept of metabolomics for some of you, which it may be new to. I guess we have to give this sort of standard slide about disclosures and conflicts of interest. I'll start off with some definitions just to describe what metabolomics is. So basically, it's another branch of things like genomics and proteomics. So it's the high throughput analysis and characterization of chemicals or metabolites. And those constitute what we call the metabolome, which is the complete collection of chemicals or metabolites in tissues or cells or organs. In the case of food composition analysis, what we're trying to do really is determine the chemical or metabolite components of foods. Most foods are from living organisms. And so in many respects, what we are looking at when we do food composition analysis is a metabolic profile of plants or animals. So the last part, I guess, is with the food biomarkers. And in this case, it's often a case of where foods are broken down in our bodies. And in many respects, we're looking at the food metabolites found in our own metabolome, our own tissues or biofluids. And the hope is that with food biomarkers, we're able to identify markers or characteristics of specific foods or diets. Now, metabolomics has a specific workflow. You'll usually start with some kind of tissue. It could be cells, it could be plants, it could be muscle tissue. And if, in most cases, what you'll try and do is metabolically quench those systems, freeze them, then you'll perform an extraction to get out the chemicals. You can avoid a lot of the extraction methods if you decide to just take biofluids, so sap from plants or blood or urine from animals. That also is a great way of getting an extraction. And then you go through and put this through a chemical analysis. You'll use mass spectrometry, liquid chromatography, nuclear magnetic resonance spectroscopy. These are characteristic analytical techniques that are found in just about any chemistry lab. And so those first 3 or 4 steps are pretty routine. The last part, which is where the fundamental change has happened, is just the data analysis. You're analyzing complex chemical mixtures, and it's only in the last 10 years or so that the software and databases and technologies have appeared that actually allow this mixture analysis to be done at a fairly routine level. And so that's the fundamental development that's enabled metabolomics. The technologies are a real alphabet soup. Chemicals are much more diverse in terms of their features or space than, say, proteins, which have 20 amino acids, or DNA, which just has 4 nucleic acids. There's probably more than 5,000 different chemical classes, and in order to be able to analyze that diversity, you need a wide range of equipment. So chromatography is essential. Electrophoresis, capillary electrophoresis, microfluidic systems are frequently used. Gas chromatography, liquid chromatography, coupled to mass spectrometry, also coupled to NMR instruments. There's also the use of infrared spectroscopy, UV spectroscopy, inductively coupled plasma mass spec, a whole range of things to be able to identify the many, many different chemical components that you'll see in biological systems. In all cases, you're looking at mixtures. The mixtures have to be separated into their components through chromatography, through gas chromatography or liquid chromatography. And this slide just really illustrates how something schematically would look. You'll run a chromatogram, you'll have some peaks, you'll isolate a peak. Under that peak, because it's a very complex mixture, there might be 3, 4 different kinds of compounds. Each of those compounds will have their mass spectra collected. And then you go and compare the mass spectra to a library. These libraries are essential, and this is essentially called spectral deconvolution. You compare the spectra and you try and match the spectra to this library of known compounds. And if you get an excellent match, then that's probably the compound. And so this can be done with both gas chromatography, MS, and liquid chromatography mass spec. You can also apply the same approach to NMR spectroscopy, and you can use the same spectral deconvolution method. In NMR, you don't actually have to run a chromatographic system. The separation is done for you by the NMR. All the compounds are separated by their chemical shifts. And this is just again illustrating where you might have a mixture of 3 different compounds, and the NMR spectra is a sum of those 3 different compounds. The challenge in metabolomics is to take the top spectrum and figure out what the components are. And you can see it's a mixture of a red and a green and a purple compound. And so that's the inverse problem. And so there are tools and software that allow you to do this now, that has made mixture identification and compound identification and quantification very easy. So in metabolomics, different tools are used with different levels of sensitivity. The least sensitive method is NMR, and typically you'll be able to detect about 100 compounds, identify them, and down to micromolar levels. The next most sensitive method is gas chromatography mass spec, and you can generally detect at the high nanomolar level, and with good equipment you can maybe identify 150 to 200 compounds. The most sensitive technique is mass spectrometry, LCMS in particular, and up to 10,000 features can be identified with the technique, and you can get down to sub-nanomolar levels. Now the problem is that when you're able to detect just about everything, at least with LCMS, many of the things you're seeing are not known or knowable, and that makes it pretty hard to publish. On the other hand, when you're working with something like NMR, most of the things that you're detecting are known, and so it makes it easier to publish. And so there's this dichotomy of wanting to be able to measure everything but not being able to publish, and being able to measure some things and then being able to publish. So the tools themselves, especially in many metabolomics labs, actually use all 3 major techniques. They are complementary. Some allow you to see hydrophobic molecules better than others. Some allow you to see volatile molecules better than others. Some allow you to see sugars as opposed to organic acids. Now, metabolomics can and is being applied to food analysis. The workflow is a little different in that instead of working with tissues from a living organism that you wouldn't eat, you're looking for tissues of organisms that you do eat. You'll look at plants, you'll look at meat, you'll look at grains, oils, milk. In some cases, if it's solid, you'll extract. If it isn't, then you'll just simply take the fluid. The extracts are a little more complex because there are different components for fatty tissues versus muscle tissues versus plant tissues and roots and leaves as you might analyze. But the same chemical analysis is performed, and in many respects, the same data analyses are performed. So in this way, with metabolomics, you're not just detecting perhaps a dozen compounds, you're detecting hundreds, in some cases almost 1,000 compounds. And this is just illustrating the comparison with sort of the traditional food component analysis, which has been around for decades, where people were generally happy with measuring the protein or ash content or some of the carbohydrates, to what you can do now with metabolomics, and obviously different instruments, but the whole range of chemicals and minerals that can be measured. So metabolomics is not just applied to health, it's applied to food composition, it's applied to food biomarker analysis. It's very rapid now. There's lots of robotic systems that are used. It's got this exquisite sensitivity, being able to detect things down at sub-nanomolar levels. If done well using isotopic standards, it can do it absolutely, get absolute quantitation, very accurate. And instead of tens of compounds, we're talking about hundreds or thousands. So the result is that there's been a bit of a rush towards metabolomics, at least as it pertains to food composition analysis. And there are even companies that are making metabolomic-based food analyzers. So Brooker is now making an NMR instrument, which you can get for about $600,000. And it will analyze wine or juice or honey now. And it'll measure all kinds of compounds. It'll be able to identify, in the case of wine, which region it's grown in, what varieties or varietals are being used. It's done for authenticity validation of juices, and same with honey. In these specialized instruments, you can measure about 50 to 60 different components in wine or juice or honey. Now, in our own lab, we've taken this concept of applying metabolomics to look at foods. I'm from Alberta. It's a large farming province in Canada. And we've taken a whole range of Alberta-grown food products from the meat, poultry, dairy, cereals, oils, vegetables, and a smallish number of fruits. And we applied, or decided to apply, metabolomics to all of these foods and characterize exactly what's in them in a quantitative way. We also did a fairly extensive literature review to look at what else has been measured or collected over the last 50 years for some of the compounds and for some of the foods we looked at. As Jason mentioned, I'm the director of the Metabolomics Innovation Center, so we have quite a number of tools in our hands. So a large number of NMR instruments, a whole range of GCMS and LCMS instruments. And so we use this arsenal of tools to try and measure what we would find in these different food products. So in the case of NMR, we would average about 50 compounds, sometimes as high as 70. In the case of mass spectrometry, between 80 and 100 compounds, about 400 different lipids and fatty acids. We did about 50 trace elements. We used gas chromatography to detect about 48 compounds. And then we had specialized assays to measure the polyphenols and lipid and water-soluble vitamins. So with this metabolomics-based multi-platform approach, we're identifying and quantifying about 600 compounds for each food item. This is kind of a hard-to-read graph, but just summarizing with the different colors, the number of compounds that we would measure for different food items for different platforms. So it's just highlighting the fact that to do metabolomics, you actually do need more than one type of instrument. In this case, they're using 4 or 5 major instruments. And for the non-lipid components, we could routinely get between 200 and 250 compounds. As an example in more detail, this is just from beetroots and illustrating the number of compounds that we can measure, usually with specialized HPLC UV assays, more specialized NMR assays, different types of GCMS assays for non-volatile and volatiles, the different metal ions that we could measure, and the variety of compounds detected by LCMS. So that information, and for all these different food products, was then put onto a database. We call it the Alberta Food Composition Database. And this is sort of picking up on this idea of open access science. All the information that we gathered and collected through this study was then put online. And it also inspired us to build out another database, which is much broader in scope than the Alberta Food Composition Database. This is just simply called the Food Database. So this one has been essentially a 5-year project where it's built on this Alberta food study, but we've been working with groups in Europe and many other labs around the world. Right now, the database contains almost 27,000 chemicals that are known to be in foods, covering about 900 different raw or lightly processed foods. So this is not the database to go and look at the chemical composition of lasagna. It's a database to look at the chemical composition of an apple or fruit or of wine. The information in the database, again, open access, has thousands, hundreds of thousands of concentration values, thousands of references. It also tracks health effects and flavor or taste data. It's able or allows you to search by structures, to search by text, to search by food classes, compound categories. It's not so much just a list, it's actually more an encyclopedia about food composition. All of it's freely available, all of it's downloadable. It's still evolving. It's consuming a lot of our efforts. And in many cases, a lot of the food compounds listed in FoodDB or a lot of the foods listed in FoodDB have up to 2,000 constituents listed for each of them. One of the reasons why we wanted to develop these databases and these resources and to apply metabolomics to food analysis was so that it could open the door to doing food biomarker testing. So if you can measure the composition about a food item, then it gives you a good idea of what could potentially be a food biomarker. a chemical in blood or urine that you could use to determine if someone has actually consumed that food. So the issue with food biomarkers, the push towards this, is because we know, or people who've been doing nutrition studies, that things like food frequency questionnaires, 24 dietary recalls, and food diaries are not infallible. In many cases, people forget or they lie. And this has made the correlations between finding food consumption and health effects quite difficult. So the hope or the intent with food biomarkers is to help corroborate or improve or validate what you're collecting from the standard methods. What we also became aware of in these studies is that most food biomarkers are not actually specific food components, they are metabolites of food components. They are transformed by your gut or your microflora or liver enzymes into something that's a little different than what you ate. And this is kind of illustrated here, which is a diagram we've used a few times to illustrate exposures, including the food we eat, and how these exposures, foods, drugs, pollutants, are transformed by our body to generate a whole bunch of other metabolomes. Food is transformed to the food metabolome. Drugs are transformed to the drug metabolome. Pollutants are transformed to the pollutant metabolome. And then there's that endogenous metabolome, the compounds that we need and produce ourselves to live and grow. So to apply metabolomics for food biomarkers, we use almost the same workflow as I showed for metabolomics or for food analysis. In this case, though, we have an intermediate step where we take the food samples and we give them to humans. And then we collect their urine and or blood, and then we analyze them using the standard metabolomic techniques. In the same way we can measure hundreds of compounds in foods, we can measure hundreds of compounds of food biomarkers through dietary intervention studies. Now, as I mentioned before, compounds we eat are typically transformed. So if you drink tea, the epicatechin that's typically in tea will be transformed by your gut microflora into benzoic acid, which then will be finally excreted as hippuric acid in the urine. If we ate meat, as most of us did this afternoon, the meat will be decomposing into carnosine, which will then produce methylhistidine, which is a marker for meat consumption. Eggs have lots of lipids. That lecithin is broken down to phosphatidylcholine, eventually to trimethylamine, and finally to trimethylamine oxide, and so on down the list. So the things on the left are essentially the foods. The thing in red is essentially the biomarkers, and many of them, as I say, are transformation products. Now, these studies with our group started about 8 or 9 years ago, but they picked up a lot of speed about 2 years ago with the development of the Food Biomarker Alliance. And this is a project that has about 20-some partners located in Europe and in Canada, and they're aligned with ILSI, Europe. And their work is to essentially produce information about food biomarkers. So the project itself has been going on for 3 years. It'll wrap up in 2018. And its goal is to use metabolomics to systematically identify and validate useful or validated strong food biomarkers. Lots of different countries, lots of different labs working on a variety of specific dietary intervention studies. Also a lot of literature reviews, which will be coming out sometime in the next 3 to 6 months, and a number of large cohort studies. So it's been funded by the European Union and by Canada, CIHR. All of the findings from this project will be made freely available, along with a number of the compounds through a compound exchange system called Foodcomics will be freely available. So some of the outputs of the Foodball project, and as well as some of the inputs, have been an example, which is the Human Metabolome database. This is a database of essentially all of the metabolites found in your body. Right now we have almost 75,000 compounds that we can find in blood, urine, and tissues. The database includes many food components and food metabolites, lots of food additives as well. And for people doing analysis, they often need to have reference spectra. So this has a large collection of referential spectra, so this allows you to identify those compounds. It's designed very much like the food database. People can do spectral and structure searches, text searches, compound browsing. They can also search by sequences, DNA and protein sequences. There'll be a major update coming out in about 2 months, which will probably quadruple the size of the database. With our French colleagues, we've also been working on Phenol Explorer. This is the largest repository on polyphenols, those antioxidants that everyone talks about. And this has been trying to collect information not only on the polyphenols found in foods, but also the polyphenol derivatives found in people, with information about their concentrations and their derivatization products, information on the food variety, commercial origin, the extraction methods to prepare the polyphenols, the analytical methods. And again, it's searchable and open access. The most useful one, at least that's come out from this work with Foodball, has been a database just released called Exposome Explorer. And this is specifically tracking known food biomarkers. This one was looking not only at dietary biomarkers, but it also tracked pollution and pollutant biomarkers. And again, it's used a combination of literature research as well as experiments done by the International Agency on Research in Cancer and groups in France, Canada, Denmark, and elsewhere. So the values here, there's right now 142 food and dietary biomarkers that they've identified, are strongly validated. They look for only or included only those that exhibited high correlations with the consumption of the food. And they provide, or in this particular database, thousands of concentration values, both in blood and urine. As I said, the correlation values are provided for all of the validated biomarkers. And like our other databases, it supports text and structure and concentration searches. So this database will grow over the coming months, particularly as the published work starts rolling out from the Foodball or Food Biomarker Alliance project. So sort of watch this space in the future if you're wanting to identify additional food biomarkers. Many of these biomarkers are valid for consumptions within about 24 to 36 hours, although some of the biomarkers are also long-term markers indicating habitual intake or will persist in the body for up to 6 months. So again, the information is in the database. So I'll wrap up here, but really I think in terms of the takeaway points, the main one is to indicate that metabolomics itself is starting to transform food composition analysis. Through metabolomics, the food composition databases have started to grow considerably. The example being FoodDB with some 26,000 compounds in foods and an average of 2,000 components in each food. The other point to make is that quantitative metabolomics, measuring absolute concentrations, is also helping to identify, quantify, and calibrate some new and very useful food biomarkers, which will be able to complement the standard food frequency questionnaires and food diaries. The other point, I think, as well, and I've tried to highlight this with the web address, is almost all of these findings are now freely available on the web. They're resources for you to use as you wish and to download and store them as you wish. They're frequently updated and will continue to be updated for as long as we have our funding. So with that, I'll acknowledge people who contributed a lot to this work. Augustin Escalbert and Claudine Manache are our collaborators in France. A list of the others are my students and staff, and then many members of the Foodball team, of which there are probably about 50 scientists across Europe and Canada, and of course the funders. So thank you very much.